So hello everyone, I'm Makoto Saito, an ECL team leader at RIKEN. It's truly a pleasure to share my experience with the HFSP and my current scientific vision. I started my own laboratory at RIKEN in this January. Before that, I worked as a postdoc with Dr. Fan Zhang. And I really appreciate the generous support from HFSP throughout the pivotal 3 years of my postdoc time. In the past, I investigated the molecular mechanisms underlying various diseases. As an undergraduate, I investigated the cellular signaling pathways in cancer cells. And during my PhD, I focused on biomolecular condensates and studied the effect of a post-translational modification on a liquid-to-liquid phase separation. By the end, I finished my PhD, I learned a lot about how disease causes malfunctions at the molecular level. As a postdoc, I wanted to find the ways to fix these malfunctions, so I became interested in the Zhang lab, where they are developing molecular tools, especially for genome editing. As you may know, HFSP encourages scientists to move into new fields. This perfectly matched my own goal to transition from molecular biology to bioengineering. Cas9 was first demonstrated for human genome editing in 2013, and when I started my postdoc around 2019, several Cas proteins and their functions were being discovered. However, to expand the genome editing tools for therapeutics, more effective genome editors were still needed. So, during my postdoc, I focused on natural diversity to identify new biological systems that could be harnessed for genome editing. These are the systems I identified and analyzed. CRISPR-associated transposon, which can insert genes directly without cutting DNA. Further, the first eukaryotic CRISPR-Cas-like protein. And Tiger-Tas, a new RNA-guided system evolutionarily distinct from CRISPR-Cas. And these systems are derived from exotic organisms. This is not the only case where biodiversity has driven innovations in the life sciences. In addition to CRISPR-Cas9 itself, some well-known examples include GFP and optogenetics. It was difficult to connect genomic information with biological function in the past. However, with the rapid progress of AI-based biology, the discovery of such useful systems is expected to accelerate. Based on this philosophy, here's an overview of my research program as an independent PI. First, I would like to focus on natural diversity and identify new biological systems. Second, I would like to characterize how the systems work. Then finally, I aim to engineer these systems to function effectively in human cells. In this way, I believe that we can develop novel molecular tools for basic research and molecular therapeutics. In relation to my job search, I would like to thank HFSP for one more thing, the flexibility of research and travel budget. In my case, I could use the budget for 6 months after 3-year fellowship time. Thanks to this, HFSP generously covered the flights and accommodation when I visited Japan. This was incredibly helpful as I was exploring job opportunities here. Last but not least, I would like to say a few words about the Fan Zhang lab, which kindly hosted me as a postdoc. Needless to say, it was an intense lab environment, but spending valuable time with some smart and dedicated colleagues there was an exceptional opportunity for me. I learned that how much dedication to science is required to make a real impact on the world. And my lab is just getting started, but one of my dreams is to establish a similar environment here in Japan. With that, I would like to close my talk. I truly hope this prestigious HFSP Fellowship will continue to support the next generations of young scientists around the world. Thank you very much for your attention.